Hello, thank you so much for joining me. I thought I'd just do a little presentation. This only consists of 19 slides, and two of them are just cover slides, the title page and the end page. I don't know how long it will take, but hopefully it won't take too long. And the reason I've done this presentation is because in my previous presentations, I feel that there were some gaps and some knowledge gaps and some missing information that maybe people might have had questions on. And as I've evolved in my learning of this subject area, I realized that there's a lot more complexity to this subject, and I just felt that there was a need to sort of explain things. So the first thing I wanted to have is this diagram, and this diagram you can get in any textbook, any immunology textbook. I just made it up now because obviously I kind of know what it is now. I don't know whether they've got all this information in immunology textbooks nowadays, but this is an overview of the hematopoietic stem cells and the different lineages. So the common myeloid progenitor cells and the common lymphoid progenitor cells. And the reason I want to talk about this is because I talked about in previous presentations about the tumor microenvironment and how problematic it can be for endogenous T cells and even also for CAR T cells. And in order to understand that, you have to understand the purpose of the other immune cells and how they impact on the CAR T cells and T cells ordinarily and how they are used by the tumor microenvironment. So, for example, yeah, macrophages here. These are ordinarily macrophages are derived from monocytes, and monocytes exist in your circulatory peripheral blood, and macrophages are differentiated into your tissue. They're tissue specific, and macrophages can be used by your tumor microenvironment to turn against the immune cells and to protect the tumor microenvironment and to protect the tumor. Macrophages also interact with your T cells. So on a normal situation, in a normal setting, when we talk about animal studies and we talk about the fact that we have to look at the persistence and the activity of these T cells, and we have to choose a model, a disease model, animal disease model that best replicates that can give us some indication of the predictiveness of the product. Yeah, if we're looking at immune compromised or immunocompromised murines, then they don't have those macrophages. We can overcome that by using a humanized animal models, but that is one of the problems, is that they don't have the macrophages that interact with the CD137. So the CD137 is the co-stimulatory molecule that exists on all T cells as well as CAR T cells. Specifically on the cytotoxic T lymphocyte cells for co-stimulation, and they're required for persistence and they're required for cytotoxic activity. Yeah, and macrophages are recruited by the T cells, that's the CD4 T helper cells. They're the cytokines that are released by those CD4 T helper cells to to help with this co-stimulation of the T cells and to keep them active and to keep them able to do their job, which is to continue to kill off whether it be the pathogen, if it's a viral infected cell, or the cancer T cells. So it's important to understand that these other immune cells are important in order to get a fuller picture of what's going on. The other thing I wanted to talk about was dendritic cells. So dendritic cells in an endogenous state, and I never really talked about this in my previous presentations because I was talking quite superficially, and I didn't really have a lot of background knowledge at that time. Sorry. But dendritic cells in an endogenous state, they produce a third signal, signal 3, which is important for a number of things. It's important for um, generating the right kind of receptors, generating the right type of T cells, the right type of T helper cells. So I've talked about signal 1 and signal 2. Signal 1 is your activating signal, that's on binding, recognition and binding. And signal 2 is your co-stimulatory, which I talked about with regards to CD28 and 41BB. Yeah. But dendritic cells also produce a third signal, which is from the cytokines, and that's required for the location of where the T cells are going to go and locate the site that requires that their actions, and for the specificity of the T cells in an endogenous setting. Yeah. Now, as I said, it's not specifically required for CAR T cells. But the tumor microenvironment acts against the dendritic cells. And I'll talk about the tumor microenvironment in a separate slide, but it's just to make you aware that CAR T cells and endogenous T cells, they don't operate in a vacuum. And the other thing I wanted to talk about are these other cells on the common lymphoid progenitor route. So we have our T cells, and we have the T helper cells and the cytotoxic T lymphocytes. We also have T cells can exist. There's two different types of T cells. I think there's well that are identified by their T cell receptors. So we have the T cell receptors that are adaptive. So this would be the I think the alpha beta chain T cell receptors, and you'll see a diagram. Oh, I don't think I've got the other one, but it doesn't matter. I'll talk about it. But you'll see a diagram of the alpha beta chain T cell receptors. But there is another type of T cells that are innate. I think they do come from the common lymphoid progenitor. I don't think I've got it on this, but it doesn't matter. I put it all under T cells, and these are the gamma delta, and these are non-specific, and they're innate, and they are located, they act as sentinels, if you like, guards, and they're located in specific regions. So they're not activated by signal 1 and signal 2. They're not, sorry, not that they're not activated, they are activated, but they're not what's the word? Specific. They're not specific. They don't target specific antigens. And natural killer cells are also important because in a normal endogenous setting, you get this under expression of major histocompatibility class 1 molecules, certain ones. And although the tumor microenvironment can overcome that, sometimes, and that's where you get successful tumors that can then evade the immune system, the natural killer cells are there to fill in the gap where if the T cell isn't able to find the major histocompatibility class 1 molecule on the cell, yeah, the natural killer cell will do the surveillance of that cell, see that there's no MHC molecules being expressed because it's been down-regulated, and kill the cell. Yeah, because the major histocompatibility class 1 molecule, that sends the stop signal to not kill the cell. And if there isn't any major MHC class 1 or HLA molecules that are expressed on the cell surface, then the natural killer cell knows to kill it. But the tumor microenvironment and certain viruses as well. I think what's the virus? I can't remember the name of the virus, but there's certain viruses that can overcome this by overexpressing a certain type of major histocompatibility class C, HLA C molecules and then overcome the fact that the T cells can't recognize them and the natural killer cells don't see a danger. And then we also have these innate lymphoid cells that produce the right cytokines and produce the right environment. And this is required for, they're innate, so they're non-specific, they're germline derived cells, but they do produce the right environment for the T cells and the other innate cells in order to do their job. And then we have the B cells that that's another discussion for itself. So the B, yeah, I don't talk about that. But so the point I'm trying to make is that these T cells interact with all of these different cells and cytokines and chemokines, which I haven't got represented here, and I might have given a list in another presentation, in order to do properly carry out their function, and they don't operate in a vacuum. Right, so this slide is just really to say to you that there are different subsets. So, and that depends upon the dendritic cells. And so signal 3, the dendritic cell will identify the type of danger, if you like, that it's dealing with, and it will then polarize these T cells, the cytotoxic T lymphocytes, as well as the T helper cells. It will polarize them to produce the right cell subset. Yeah. So there's things that I've talked about in the past. I haven't talked about this, but I've talked about phenotypes. Yeah. So there are different phenotypes. For example, the stem cell memory T cells, the central memory T cells, the effector T cells, effector memory T cells, etc. Yeah. And then there are these subsets, and these subsets are specialized, if you like, to carry out a specific kind of response depending upon the target that we're looking at. So, for example, intracellular and bacteria viruses are different to, say, for example, worms and other protozoan parasites. So the dendritic cell is important in an endogenous setting to identify, or not to identify, but to drive the correct subset that's required. Now, when we're looking at T cells, CAR T cell engineering, and we're looking at the autologous type, and I presume also, although I haven't checked, the what's it called, the T cells that will be based from allogeneic sources, so that's stem cell derived and differentiated T cells, yeah, that will be CRISPR-Cas engineered appropriately so that it can be an off-the-shelf product, yeah. The TC1 is the specific type that is to be selected. TC9, I don't think that there's a lot of, I think it's recent, and I don't think there's a lot of information, or at least I haven't yet looked to see. So the other thing I wanted to talk about was the cytokines. So IL-2 is an important cytokine for cytotoxic T lymphocytes, CTLs, yeah, but it's produced by the T helper cells. It's not produced by not much is produced by the T cells themselves, because they are the main consumers of this particular cytokine. But they do produce interferon gamma and tumor necrosis factor alpha. Interferon gamma is supposed to, these cytokines also promote macrophage activation, similar to the T helper cells. I'll show that in the next slide. But interferon gamma causes the cells to start to express the major histocompatibility complex in order for the T cells to attach themselves to, and in order for the T cells to even identify. And tumor necrosis factor alpha is responsible for opening up the area so that the monocytes can differentiate into macrophages and they can reach the site of interest in order to carry out their phagocytic, cytotic, I can't remember how you say it, function. So the key cytokines are interferon gamma and tumor necrosis factor alpha. So the T helper cells, now these are really important. It's important to know about these because they help, if you like. They provide the right environment, so they provide the interferon gamma, the tumor necrosis alpha. The T helper subset 1 is what we're really interested in, because it's specific to viruses, intracellular bacteria, but also for tumours as well, cancers and tumours. So they provide the right environment, and they help the T cells by producing the right cytokines for the cytotoxic T lymphocytes, the CD8 plus cells to operate in. Again, it's also important to have an overview of these other types because of the fact that these T helper cells are plastic and they can convert to other forms. However, the T follicular helper cells, they're much more specialized form of T helper cells that are formed with the dendritic cells interaction with T helper cell, naive type T helper cells. But anyway, Treg is really important for us to know. We need to, number one, ensure that there is minimum amount. It's an impurity, if memory serves me correct. But also Treg turns off the cytotoxic T lymphocytes and it kills the cytotoxic T lymphocytes. It consumes all the IL-2, which is required by cytotoxic T lymphocytes. It secretes IL-10 and TGF-β, and it acts on all the cells. And those two cause so much problems because that's part of the tumor microenvironment where the tumor cells recruit Treg and you know, they use Treg to shut down the actions of the T helper cells and the cytotoxic T lymphocytes, so the CD4 plus and the CD8 plus. So it's important to know about these subsets. I can't recall if I've said it. So Treg also converts Th1 into Tregs, as you know, which then perpetuates the vicious cycle. So that's the reason why I've got this presentation slide. I can't remember. I'm pretty certain I didn't put this in my presentation previously. So I just want to talk about immune homeostasis because it's important to know. It's an important what's the word I think step that's required, and it's basically required when the immune response is no longer needed. So normally if you have a viral infection or damage or cancer tumor, you get an immune response. So you have this normally occurs, then you get an immune response. Hopefully, you know, it's a successful immune response. You deal with the infection, the damage or the cancer. Damage doesn't get a strong immune response. You only get a strong immune response if you have damage associated with an infection, because you don't want to trigger a strong immune response every time you get a damage, you know, because you don't want that. So the immune system is what's the word I'm looking for? Evolved to not respond strongly to damage only, but to respond where damage is in association with an infection of some sort. But where you have these three actors, if you like, you get an immune response, and that immune response is good, but you want it to stop once these infections and this damage and this cancer or tumor has been treated and has been addressed. And that's the purpose of the immune reset, where you have your, and this is what we refer to as immune homeostasis. So you want this balance to occur. You have this balance that occurs normally when everything's fine. You have an infection, then you have your immune response, and then you want to bring everything down to back to the balanced state that it was in for this immune reset. And this is the immune homeostasis. And the reason is all these pro-inflammatory cytokines can cause damage. You know, when you have an immune response, you get swelling, you get mast cell swellings, you get edemas, you get inflammatory molecules, you get reactive oxygen species, etc. You get all kinds of things that are great for killing or dealing with the trigger, but you don't want it there permanently, because it will then start attacking healthy cells. And that's why you need this immune homeostasis. And that's the problem, because the tumor microenvironment exploits this immune homeostasis, this anti-inflammatory cytokines, and it uses it against to switch off this pro-inflammatory cytokines and the immune response. It uses it to switch it off, and it means that you can't deal with the cancer or the tumor, and the tumor evades the normal human immune system, and also evades in cases of solid tumors, it can evade the CAR T cells. And so there are mechanisms that we have in order to deal with this. So this slide, it's a bit complicated, and apologies for that. I was a bit busy, but really I just want to talk about Tregs and the impact that it has on everything, and how it really can, how the tumor microenvironment employs Tregs and what the impact is. And Treg is necessary and important, as I've said previously, in that it is good for this immune homeostasis. However, it has a detrimental effect when it's used by the tumor microenvironment to fight off our normal immune response that helps us to deal with this tumor. So Tregs, oh, have I got it there? Yeah, it is. Here it is. Tregs produce IL-10 and IL-35, and it also produces TGF-beta. TGF-beta converts TH cells to T helper cells to Tregs. Yeah, so that is a vicious cycle going on there, because you don't want to do that. You want to, the T helper cells are required in order to produce IL-2. And the IL-2 is required for the cytotoxic T lymphocytes. And so you basically switch off and turn off, and the cytotoxic T, the CD8 plus cells are not able to kill, because they don't have the fuel, they don't have the cytokines, they're not in the right conditions. TGF-beta also turns off the cytotoxic T lymphocyte and the T helper cells. It turns them off. It mutes, so it mutes the TNF alpha signal and the interferon gamma. So no more warning signs, no more inflammation. So it mutes that, and it converts TH1, which is the helper cells, into Tregs. Yeah, which then secretes more TGF-beta and more IL-10 and IL-35 that has a an even more sort of vicious cycle, detrimental impact on the inflammatory response that we want for this tumor. IL-10 stops the dendritic cells from doing that signal 3. Treg cytokines bind to the cytotoxic T lymphocytes, yeah, and they block the transcription that results in the transcription of the genes that results in TNF alpha and interferon gamma. Yeah, so it basically turns everything off, which is great if and when you've got inflammation, and you've got you've got an infection, and the infection has been dealt with, but the tumor microenvironment hijacks it. It creates an ecosystem where these Tregs are doing this. M1 to M2, these are the macrophages, are converted to M2 in that environment, and M2 is anti-inflammatory, and that pumps out even more TGF-beta. TGF-beta, which then converts more T helper cells into Tregs, and also TGF-beta binds with arginase, or I can't remember, but arginase is required. Arginase is an enzyme, and what it does, it works normally I think it's in the liver, but in the tumor microenvironment, it basically uses up arginine, and T cells need arginine to build their CD3 zeta chain. So I'll talk about that in some of the next slides. Yeah, so it works against the whole of this microenvironment, works against the body's immune system. You upregulate CTLA4, which competes with CD28. Now, that's not a problem for CD28 is the co-stimulatory receptors, but that's not a problem for CAR T cells. But in an endogenous setting, that is a problem, because CD28 is required for co-stimulation. Yeah. And I've got it here, DC dendritic cell maturation signal 3, and it also inhibits CTL activation. Yeah, I think I've discussed that. And also Tregs are cytotoxic to cytotoxic CD8 plus cells, so they will kill them off, because obviously if and when the whole thing of the infection or the tumor cells or the danger has been addressed, you don't want any more T cells there, so they'll kill them off. So this is how the Tregs is also, I mean, I put it around Tregs, but it could be TGF-beta, any one of these. All of these are equally important. This is how the tumor microenvironment interacts with the different T helper cells, with the different cells, in order to evade the immune system and, become a big major problem. IL-10, as I said, it prevents the signal 3 from the dendritic cells, and without this signal 3, you can't have that proper activation. Yeah, so this is really important to talk about the impact of all these other cells and all these other cytokines on CAR T cell and on the endogenous T cells. Right. So for this diagram, I think I wanted to talk about this because I talked about ITAMs, and I never really talked about CD3. I never really talked about it in a lot of detail, and I just felt that I should really give a bit more explanation. So T cell receptors, this is the adaptive type, so it's an alpha beta heterodimer. Yeah, so the other type, which is the gamma delta heterodimer, that's the innate T cells, and they're very small in numbers, and they tend to exist in certain locations in the body, in certain strategic locations in the body, such as the skin and the gastrointestinal tract. But sorry, this whole system requires CD3, and CD3 is on all T cells. So it's a specific complex, a specific marker for all T cells. That's how we identify them. And remember, CD8 plus is identified by the CD8 co-marker, co-receptor, and CD4 plus by the CD4 co-receptor. But we're not talking about that for the time being. But I just want to talk about this because I did talk about ITAMs, and I did talk about, the fact that some what do we want to call them? Some research and some clinical trials involve reducing or knocking out some of these ITAMs on the CD3 zeta homodimer, in order to reduce the intensity, if you like, of the signaling, and to help with regards to persistence and reduce the potential for cytokine release syndrome, so the safety issues. So ITAMs, as I said before, well, these are immunoreceptor tyrosine-based activation motifs, and they are basically, they're like launch pads. Once they're phosphorylated, you then get other molecules that are required for the activation of the T cell, and downstream it results in the immune immune synapse, and the release of the lytic payload into the target cell. But CD3 consists of these three. That's a good thing. Nice. It consists of these three components. It consists of two different heterodimers. So it's the epsilon delta heterodimer and the epsilon gamma heterodimer. And each of these have an ITAM. And then it consists of one homodimer, which consists of the zeta homodimer. Yeah. And this has 3 ITAMs each. So there are 10 items in a normal endogenous setting. There are 10 dimers first, yeah? And the CD3, the whole thing is a T cell receptor complex, which consists of the CD3 molecules. But the T cell receptor complex, it doesn't go down deep enough intracellular, deep enough, and it relies upon the CD3 structure, which is invariant. Sorry, the CD3 is invariant, it doesn't change. And what CAR T cell does is it uses that invariant structure to sorry, I'm using my hands to activate. So it's still using the CAR T, the T cell structure, but instead we've replaced the T cell receptors with the CAR. So let me just move to the next slide. So here, I just want to talk about something else. So when I talk about CRISPR-Cas engineer, I talk about knocking out the track, the T cell alpha constant. So that's this, yeah. And once you knock that out, you don't need to knock out the T cell beta constant, because it's an obligate dimerization. Without the alpha, this will fall away, so you don't have a problem. So that's what I mean by that, and that's why, as I said, we knock out the T cell alpha constant region on the gene, and once you've done that, you don't need to knock out the T cell beta region on the gene. And you need to think about being efficient with regards to your CRISPR-Cas engineering. If you don't need to knock something out, don't bother. There's a lot of changes that we're already making. So we have to be mindful of doing any additional changes, particularly where there's a risk of unintentional gene editing. So that's that explanation for that. And yeah, I've got here ZAP70. So ZAP70 is a signaling protein, and this ZAP70 is required for launching onto these ITAMs, and downstream you get the what's the word I'm looking for, the effect of the T cells, which is for them to release their lytic payload into the target cell. So that's what I haven't talked about, and I just assume that everybody has an understanding, but it's just that every time I read papers, I'm reading about these heterodimers, and I don't know whether people are reading any papers on this, any scientific journals on this, but if you do, you'll see these references, and you do need to understand what you're talking about. If there's anything else I should talk about, let me have a look at my notes. Yeah, I don't know if that's but basically these items get phosphorylated, yeah, by certain kinases that add the phosphate group, and to the tyrosine residues, and once they're phosphorylated, they then attract the ZAP 70. And this whole case of phosphorylation requires ATP, adenosine triphosphate. The phosphate group is released, and that requires glucose, not sugar, requires glucose and glycolysis. And that's really something that's important to know, because in a tumor microenvironment, there is a deprivation or depriving of glucose, which is used by the tumor cells, and the T cells don't have the fuel, if you like, in order to activate and in order to kill off the T cells. So the tumor microenvironment acts in a way that is detrimental to the T cell. This is just showing, in the case of major histocompatibility class 1 molecules, showing the interaction with the T cell receptor. But the same thing happens. This is for the alpha beta heterodimer receptor, which interacts with the major histocompatibility class 1 molecules. For in the case of CD8, yeah. And the same thing happens, yeah. And this is just giving you, just a pictorial representation of the CAR construct. Now, I've just put the one receptor, the CD19 receptor, that binds to the CD19 on the B cell. So the CD19 is the B cell antigen, and the CD19 receptor on the CAR, that's the CAR, chimeric antigen receptor on the CAR. I should have put CAR receptor, really, never mind. And the CD28 or 41BB, that's intracellular, so we don't need that. So in the tumor microenvironment, we'll normally overexpress CTLA4, I think it's called CTLA4, which competes with CD28 for co-stimulation, but we've sorted that problem out by putting it all together. So basically, the point of this is to show how the chimeric antigen receptor utilizes the architecture and the structure that's already present in the T cells in order to achieve its objectives. Any notes? Oh, I don't know whether this is the right place to talk about it, but when you get binding, you get the release of certain markers, and those markers can be tracked with flow cytometry. When you get degranulation, they're degranulation markers, and they can be detected by flow cytometry. So one of what we have to do when we're looking at the potency is we take an orthogonal approach, where we're looking at a number of things. So we're looking at, say, for example, the release of cytokines. We're looking at the markers that are that we can use to demonstrate the state of the T cells, i.e. activation, i.e. degranulation, the release of the lytic granules, all of and also the killing of tumor cells. So we can use T cells, put them in the presence of tumor cells, and measure over a period of time the reduction in the tumor cells, and look at the potency and activity of the tumor cells. So this is how we do it, basically. This is what happens, but this is what we are further downstream in terms of the development and the thinking process. In terms of when we're looking at our potency, that's what we're doing. Right, I just want to quickly talk about the tumor microenvironment, because it's really important when we're dealing with solid tumors, and this particular project is looking at CAR T cells, looking at using them for solid tumors, and what we are dealing with, and what we have to overcome. Oh, what's the matter with my click? Why isn't it working? Oh, there it is. So your tumor microenvironment is like an ecosystem, and it is a hostile ecosystem because it uses the immune homeostasis mechanism that the body naturally has against the inflammatory response that the body requires in order to kill the tumor. So in a sense, it's a hostile ecosystem. It's hostile to what we want, which is we want pro-inflammation, and the tumor microenvironment is anti-inflammatory and recruits the immune cells in order to, I don't know whether I talked about that in that diagram, but it recruits the immune cells to work against the pro-inflammatory molecules and cells that will kill the tumors. So it's a hostile ecosystem. It works in three ways. Whoops. To stop, to evade the natural endogenous and also engineered T cells. So the first is it's acidic and it's low in oxygen. So lactate is a problem, and adenosine, which basically turns off the T cells. So the T cells require oxygen in order to have this phosphorylation in order to operate, and if it's low in oxygen, hypoxia, then that's a problem for your T cells. Yeah, and another important thing is glucose and arginine. So I don't know whether I did mention that arginine is required for the CD3 zetas, and it manufactures arginase. L-arginine is required, and it manufactures the macrophages manufactures, I think it does, L-arginase, which consumes all the arginine. Yeah, and therefore your CD3 zeta, which requires arginine, can't operate. Yeah. CD28, when it's used in the CAR T cells, or if it's used in endogenous T cells, they are high energy consumers. So they require glucose via glycolysis, yeah, in order to phosphorylate those CD3 zetas, yeah. And in order to do that, they require glucose. And if there's no glucose because the tumor cells is using it all, because the tumor's cancer cells, they use up a lot of glucose, the CAR T cells themselves end up in a state of metabolic stress, yeah, because without glucose, the ATP, the T cells cannot generate the ATP cells that are needed to phosphorylate the CD3 zeta tails, and consequently, the CAR T cells are hit at when this happens, because the CD3 zeta stays cold, and there's no chemical energy for phosphorylation to occur. So this is really a real problem. And the other thing about the tumor microenvironment is its overexpression of PD-L1. So PD-L1 is the ligand, and PD-1 is the receptor on your T cells, and it basically switches off the receptor on your T cells, and therefore prevents your T cells from being activated. And this is a problem because this overexpression means that your T cells can't do anything, and doctors are now giving checkpoint inhibitors as well as a way of treating cancers, but that is one of the tumors, and that is one of the problems of the tumor microenvironment. Yeah, so you will often see this terminology, this MDSCs. Oh, should be small s there, but doesn't matter. Myeloid-derived suppressor cells, and M2. I've talked about the M2 macrophages, where M2 is converted to M1 is converted to M2 in the tumor microenvironment, a suppressive environment. So you'll often see these myeloid-derived suppressor cells. Oh, sorry. And the problem is that they have a counter effect on the immune system, on the T cells. So macrophages are pro-tumor. So they will secrete IL-10 and TGF-β, which downregulates the T cells and converts them into bystanders, and basically tells the T cells to stop fighting and to start resting. Yeah. And it will convert the T helper cells into Tregs, which then perpetuate the situation as I've discussed previously. Oops. And the other one is tumor-associated neutrophils. So I talked about neutrophils in that one of the innate phagocytes that are responsible for phagocytosis of I think it's extracellular bacteria. But in the tumor microenvironment, they flip to a specific type, an N2 phenotype, and instead of killing bacteria, they release their reactive oxygen species, which then damages your T cell membrane. So this is what these are the myeloid-derived suppressor cells. There's some other ones as well. So these are the cells, yeah. But there's some other ones as well that the tumor microenvironment employs in order to fight the natural endogenous immune system and immune response by recruiting the immune homeostatic or the immune homeostasis against the immune cell and basically turning off the immune response when it's needed. I also want to talk about fibroblasts because they provide the physical barrier that makes it impossible for CAR T cells to actually get to the tumor cells. And they basically are derived from mesenchymal stromal cells, and by nature, by their characteristics, they have anti-inflammatory properties, yeah, and they secrete anti-inflammatory collagen and components that mean that the CAR T cells, I don't know whether I put them down, but yeah, I think I did in the next, yeah, that means that the CAR T cells can't actually physically get to the tumors. And yeah, they secrete a chemokine, CXCL12, and these trap the T cells in the stroma, so they never actually reach the cancers, and they also produce TGF-β, which is a master suppressor. I just want to quickly talk about the different types of tumours and the terminologies that's often used in papers that I read, hot tumours and cold tumours. So hot tumours, these are where we can actually get, the body actually recognises that they're tumours, they're not being switched off, and it's easier for the CAR T cell to join in the fight, basically, because they're already full of T cells, the danger signals are already there, and they're already releasing cytokines as well that advise that the tumor is there, yeah. And then there's cold tumors, and these are like immunological deserts. Basically, there's no T cells. The tumor microenvironment is so suppressive that the CAR T cells just don't even get there. And it's so thick and suppressive. And these are seen in certain cancers like pancreatic cancers. So just in case you see that word, because I see it a lot. I want to talk about the engineered TGF-β receptors that I've talked about in the past, because there's an update that I want to make. So there are different types, or two types that I've identified. There's the dominant negative receptor, and this is where you basically take away the ability of the TGF-β to actually signal to stop. So you have the receptor there, but you have no signaling mechanics. It's just been disabled or removed. I don't know how to do it. I haven't looked into that in detail. And it just means that the TGF-β can't work. It can't send the stop signal. So it basically soaks up all the TGF-β in the area. And the tumor microenvironment has a lot of TGF-β, so it's a lot of soaking up to do, I guess. But it doesn't prevent the T cells it doesn't convert them to T helper cells, or the T helper cells, so Tregs, and it doesn't switch off the T cells, so it doesn't perpetuate this vicious cycle where the pro-inflammation that we want is being switched off and converted to anti-inflammatory. And then, oh, I've meant to put receptors there. And then we have the inverted switch. So the inverted switch is where the TGF-β, which is the ligand, if you like, activates the T cell receptor, the TGF-β receptor on the T cells, yeah? So instead of telling it to stop, it tells it to go. Now, this is the difference where I've talked about in the past. In the past, I've talked about using CD3 or CD3 zeta switch or CD28. And from what I've found, they did try to wire TGF-β with CD3 zeta and CD28, and it does work, but it only provides a signal when the T cell is already touching the tumor. And if the T cell can't get to the tumor, then it won't provide that signal. Yeah. So they've used, or the proposition is to use the IL-7 and toll-like receptor. So TLR stands for toll-like receptors, and they are basically innate receptors that are found on the myeloid cells, such as neutrophils, dendritic cells, phagocytic cells like eosinophils and basophils, etc. Yeah, and they use them because these signals are survival. And if you wire IL-7, the TGF-β environment acts like a constant growth hormone, so it keeps your T cell alive and young, even when the T cells are not currently fighting the cancer. So it's a better option than to just use the CD3 zeta or the CD28. That's what I found, and that's what I'll be going with. And yeah, so the overall result is when you rewire it and you have this inverted switch, is that the tumor microenvironment, which usually tries to suppress the CAR T cells with the TGF-β, activates it instead, and this works in favor of CAR T cells. I just want to talk about CTLA4 and CAR T cell architecture, and this is just a brief slide because it's not really relevant for the CAR T cells, but it is relevant for the for the normal endogenous T cells, and that is that CTLA4 is a decoy receptor. It's a normal thing because you don't want T cells, you don't want that overactivity. So it is much more stronger binding to the B7 proteins. That's the CD80 and CD86 proteins that are exhibited on dendritic cells or other cells, say, than the CD28. And what happens is, in a normal situation, the tumor cells, it overexpresses CTLA4, and that means that the T cells won't receive in the endogenous situation, it doesn't receive the signal, the second signal for co-stimulation, so it steals it away from it. And it's even normal, it competes because you don't want overactivation. You want to have a controlled immune response. Yeah, but the cancer will, what's the word I'm looking for? It will overexpress the CTLA4, and this is a checkpoint inhibitor, and there are a checkpoint, and there are checkpoint inhibitors to bind with this in certain cases if you're taking certain therapeutic strategies. However, for CAR T cells, because we've moved the CD28 or the 4-1BB inside the intracellular region of the cell, you don't need the B7 proteins, so it's not a problem. But the other problem that's still there is the PD-L1. So the PD-L1 is the ligand that binds to the PD-1, which is the off switch on the T cell receptors, and that will cause a stop. And this is the reason why many patients receive CAR T cell therapy along with checkpoint inhibitor drugs that bind with either PD-1 or PD-L1 to prevent that off switch. So, there's a lot to it. Now, I want to talk about natural killer cells because ordinarily, when you have this downregulation of your major histocompatibility class 1 molecules on your cells by the cancers or sometimes by viruses, your natural killer cells will come in along and fill in that gap, see that there's no any major histocompatibility, recognize that there's been an infection or there's a tumor or cancer, and kill off the cell. Yeah. So normally, when the tumor tries to hide the major histocompatibility by downregulating, the natural killer cells would see the missing self and kill it. Yeah. However, the problem that we have is that some tumors and some viruses, so let's talk about HLA-E overexpression. So they will talk about I've talked. This is something that I've talked about, but they will talk about they will overexpress a certain type of HLA-E, which will bind to the receptors of the natural killer and provide that powerful do not kill signal. Yeah, and some viruses, they secrete the major histocompatibility class C, MHC-C, which is a different subtype. I can't remember which one it is, but some viruses do that, and therefore they evade the natural killer cells that patrol and look for the fact that there's no major histocompatibility to kill the cells. So viruses and tumors can overcome or evade our normal immune system and prevent the cells from being recognized and killed off. In addition to that, some tumors can, so MICA stands for major histocompatibility class 1, A related A proteins and B proteins, and they're expressed on the surface of the cells. Yeah, and these natural killer cells can see these major histocompatibility class 1 A and B proteins and bind to them, and therefore it's not a problem. But some tumors can shed them in the fluid, and the natural killer cells then attack those receptors or see those receptors, and their attack receptors bind to those receptors, and they don't perceive a danger. So as I said, the major histocompatibility class 1 are the ones that the natural killer cells patrol, and they bind with these, and they send the don't kill me, the don't kill me, not what's it called? It's called killer inhibitory receptors. Yeah, so they have a strong signal there, but when they're not there, you've got the killer activation signal receptors that are activated through ubiquitous molecules on the cells. So under stress conditions such as viral infections or DNA damage or heat shock, the cell normally ramps up these major histocompatibility class A and B molecules and moves them to the surface. Yeah, so that they can see that there's a problem. And also the TNF, interferon gamma also has that effect as well. But in tumors can shed those proteins, and they bind with the natural killer cells, and they can have a thing. The other thing is, have I got that here? Yeah, TGF-β has the same impact on natural killer cells as it has on T cells, and so it prevents the natural killer cells from producing the lytic proteins and releasing their lytic, the degranulation effect. It prevents that from happening. So the tumour microenvironment, it hijacks the immune homeostasis system, if you like, where it hijacks the immune system for its own benefit. And, you know, there are all kinds of, in addition to what I've explained, there are all kinds of things that the tumor microenvironment does that I haven't discussed here. It's a separate conversation in itself. But it's important to recognize how the immune cells, how they all work in concert with one another, and how they, what's the word I'm looking for? How they are all influence each other in terms of their ability to attack, their ability to persist, and to have that continued therapeutic effect. So that's it. I think it's 1 hour exactly. Thank you so much for my listening to my presentation. I hope you've enjoyed it. If you have, please don't hesitate to leave a like or a comment, and I hope to see you again next time. Thank you. Bye bye.